I need to use only the verified facts provided. Danakil is one of the lowest and hottest places on Earth, with land over 100 m below sea level, average temperatures among the highest recorded, neon acid pools, salt flats, and microbes surviving there. Scientists study it as an analog for Mars or early Earth. Sunlight fractures on the surface of a sulfuric pool. The water glows electric green, thick with dissolved salts. It steams constantly. Air shimmers above the salt flats, reflecting the relentless sun. Temperatures climb beyond 50 degrees C. This is not a place for life. It is a chemical wound on the Earth's surface. Then a scientist lowers a sterile sampler into the acid pool. The device returns, collecting a barely visible film on its surface. Back in the field lab, under magnification, dots move. Microbes, not just surviving, reproducing, in water with a pH near zero, saturated with toxic metals. The evidence is microscopic, undeniable. The discovery wasn't a single moment. It was years of sampling, filtering, analyzing samples from the most extreme pools. Scientists confirmed the microbes are extremophiles, using chemical energy, not sunlight. They found similar life forms in other acid pools across the depression. The question isn't whether life exists here, it's how it does it. The implications ripple outward. This isn't just about Earth's most hostile environment, it's a blueprint. The conditions mimic early Earth before oxygen or the surface of Mars. If microbes thrive in Danakil's acid, they could potentially persist beneath the surface of Mars. The evidence is in the pools. The mystery deepens. What specific mechanisms allow this survival? The answer remains buried in the neon glow. The pools glow under the relentless sun, a toxic ballet of neon green and orange. Sulfur boils in shallow basins, releasing the sharp scent of rotten eggs. Salt crusts crack like shattered mirrors across the ground. Scientists confirmed it in the lab. Microbes thrive here, not by photosynthesis, but by consuming sulfur compounds in the acid. They extract energy directly from the rock's chemistry. No sunlight penetrates these depths. No oxygen is required. The samples hold undeniable proof. Life persists where thermometers read 50 degrees C and pH levels plunge to zero. The evidence is not abstract. It's in the vials, the microscope slides, the data streams. One theory hinges on metabolic versatility. These microbes don't rely on a single chemical pathway. They switch between using sulfur, iron, and even hydrogen gas as energy sources. Researchers observed this by tracking sulfur concentration changes hour by hour. When acid levels spiked unpredictably, the microbes adapted within minutes. A single strategy would collapse under such volatility. But this flexibility, this ability to pivot between energy sources, creates a survival net. It's not guesswork. The lab data shows metabolic shifts correlating directly with environmental shifts in the pools. Another explanation centers on physical armor. Microbes form dense, sticky biofilms woven from their own exudates. These films coat the microbes, creating a buffer against the corrosive acid. Salt crystals growing around them may offer further protection. Under the microscope, scientists see salt lattices embedding microbial clusters. Within these crystalline cages, pH stabilizes slightly, temperature moderates. The salt isn't just a byproduct, it's a refuge. The evidence comes from samples where microbes were found exclusively within asterisk salt crystals, never free-floating in the open acid. The strongest case lies in their genetic blueprint. DNA sequencing reveals unique genes coding for acid-stable proteins. These proteins resist unfolding in near-zero pH, maintaining function where others disintegrate. The genes show clear evolutionary signatures, mutations specifically selected for extreme acidity. This isn't random chance. It's a targeted adaptation forged over millennia. The genetic evidence directly maps to the survival mechanism. The answer isn't hidden in the environment. It's written in the microbe's own code, waiting to be read in the neon glow. The case remains open, but the evidence now points decisively toward molecular adaptation as the core solution. Biofilms coat the microbes, a sticky shield against the acid, but the pools remain liquid. The microbes don't just sit on the surface. They vanish into the salt crystals growing beneath the acid. The biofilm isn't the entrance. It's the first layer of the refuge. The salt lattice forms asterisk around asterisk them. This suggests the microbes actively seek the salt, not that the salt protects them passively. Genetic evidence confirms their proteins withstand near zero pH. These adaptations are real. Yet the genes alone don't explain how they move from open acid into the protected crystals. The DNA tells us how they function inside the refuge, not how they enter it. The genetic blueprint solves part of the puzzle. It doesn't solve the entry mechanism. The refuge exists. The microbes are inside it. The journey remains unseen. Direct observation would settle this. We need to watch the microbes interact with the acid and salt in real time. Do they swim toward crystallizing salt? Do they secrete a substance that triggers crystal formation around them? Current samples show only the end result, microbes embedded within the salt lattice. We lack the video of the process itself. Without witnessing the transition, the case remains incomplete. We know the environment is hostile. We know the microbes survive within the salt. We know their genes are tailored for the acid. But the critical step, how they get from the acid into the salt, is missing from the record. The evidence points to a coordinated behavior. It's not passive, it's active. The missing piece is the mechanism of entry. The case file is nearly closed, but one crucial detail is still sealed. How do they get inside?